I'm delighted to be here. I promised Professor Brown that I would email him some photos of the cherry blossoms in Washington, D.C. And if you'd like to have a copy of those, email him and he'll forward them to you. My talk tonight is focused on ethical issues raised by new discoveries in stem cell research. And I want to just do a brief review, maybe illustrate what Professor Brown has been talking about, which is going to be kind of hard because this is not forwarding, guys. Do you want to give me the next slide? What am I doing wrong? Here we go. So stem cells are known as the master cells of the human body. As Professor Brown said, they are unspecialized cells, sometimes people call them blank cells, that can divide and renew themselves almost indefinitely, at least for long periods of time. And they have the ability to give rise to specialized cell types of the human body. This is known as differentiation of the cells. So these are unique cells that no other cell in the body can imitate. Could I have the next one, please? Here we go. This is a photo of an adult stem cell from human bone marrow. I was talking to some people at dinner tonight. Mesenchymal cells can be derived from bone marrow, and there's studies going on now to see whether these can be used to treat patients who have had heart attacks, whether they can be inserted into their hearts and differentiate into cardiac cells that proceed to fill in the gaps that have been caused by their heart attacks. There are all sorts of other kinds of adult stem cells, skin, fat, and they're found in, not just in human adults, they're found in fetuses, umbilical cords, and placentas. It's working now. Human embryonic stem cells are derived from human embryos, and these are embryos that are about 4 or 5 days after fertilization. So when people link this question to the abortion question, they're not quite right, because obviously the abortion question arises much later. And the main focus of concern with regard to human embryonic stem cells is the fact that they're derived at this early period. Most of the embryos from which they are derived were created for in vitro fertilization by couples who were seeking to have children. So the embryos were originally created for reproductive purposes. They are outside the body. Most of these embryos are frozen, although some fresh embryos are also used. And by and large, these are embryos that could not be used in the first round of in vitro fertilization. There are more embryos created than are needed so that the woman can avoid having to go through the process of hyperstimulation of her ovaries to produce eggs more than once. This is a somewhat risky procedure, very uncomfortable for many people. And the idea is to have these frozen embryos in storage so that if the couple wants to have a second try at IVF, they can use these frozen embryos. And they've had some success in having children derived from frozen embryos. Some of these embryos are of very poor quality and would not be used in future attempts at IVF because of that. But interestingly, they have developed human embryonic stem cells from embryos of very poor quality, be they frozen or fresh. So these embryos, here you see one in vitro in a glass dish. It's an expanded blastocyst. The embryos we're talking about are at the blastocyst stage. You can see the circled inner cell mass where the embryonic stem cells are derived from. This is what an embryo would look like at 4 or 5 days inside a woman's body. This is vastly enlarged. The size of the stem cell, the size of the embryo would be even smaller than the dot of an eye. So basically, what they do in order to derive these stem cells is they dissolve the trophectoderm, the extra embryonic layer of the embryo. Then they go into the inner cell mass and remove the stem cells from the embryo, and this does destroy the embryo. Here you see some embryonic stem cells that have been derived this way. Well, basically, this all raises questions about whether the embryo is a human individual that should be protected from destruction. And some would argue it should be whether it's inside a woman's body or outside a woman's body. These stem cells would be used for therapy. Here you see a sperm and egg united to produce a fertilized egg. From there, it develops to the morula stage and then the blastocyst stage, which is the stage at which embryonic stem cells would be removed. You can see then that the inner cell mass from where they're derived, the stem cells are taken, put into a glass dish or whatever laboratory equipment is available. Various methods are used to differentiate them into a variety of specialized cells, and hopefully the differentiated cells could be used to treat patients with various kinds of illnesses. Well, the basic question of philosophy and public policy clearly is, what do we think about the moral status of the embryo? And you've had philosophers, you've had politicians come in and tell you what they think about the whole situation. During recent years, President Bush in 2001 established a policy that allowed the use of some embryonic stem cell lines. And these lines had to be derived before August 2001, the date of August 9th, I believe it was, the date of his speech. The idea being that this would not encourage people in the future to derive embryonic stem cells from embryos with public funds because they would know that there would be no public funding for it. The damage had already been done in the past, in his view, that it was damage. And so he felt that this at least opened the door to starting stem cell research with federal funds, and yet would not encourage people to pursue it by destroying embryos in the future. Now, there are 22 stem cell lines available out of what were originally considered to be some 70-odd stem cell lines that could be used. And the difficulty with these is that they were fed with mouse feeder cells, and there is concern that these mice cells would carry viruses that, if transplanted to humans, could cause damage to them. They are getting old and degenerating. It's unclear whether genetically they could be used to match patients. So there are a host of concerns about using these stem cell lines. And scientists are attempting, and with some success with private money, to derive new human embryonic stem cell lines that would not use animal feeder cells that could be safe to use in human beings. And of course, the question that arose was the moral significance of the early human embryo at approximately day 5. Is it wrong to destroy this embryo? Now, various groups and individuals have adopted different views of the moral significance of the embryo. What I'd like to do is go through them with you, trying to give each view a fair shot. I can't be as complete as I would like to in our limited time together, but why don't we give it a try and see how it goes. There is the time of fertilization view that says that the embryo is a living human being at the time of fertilization, And basically, the reason for this is that you have the human genome in this one-celled entity. And because of this, the argument is it must continue to develop as an individual human being in ways that are fixed by those genes. So the idea, too, is that embryo polarity, the development of an axis, a north-south axis, can be perceived at this time. And this indicates that it is an entity that is going to proceed in a certain way. That north-south pole will continue on through the development of the embryo, indicating that it's always one and the same individual. And if some scientists in the group want to interpret this, I put this in here for you. This is derived from an article by Magdalena Zerika-Goetz, who is at Cambridge in the UK, and she is somebody who has carried out studies that argue for embryo polarity from day one. So this, and this is just to give you a sketch, an outline. The basic conclusion that people who hold this view come to is that scientific information, along with metaphysical considerations, indicate that the early human embryo that's featured in human embryonic stem cell research is an individual living human being from day one. Now, there are those who would say that this is much too early a period of time in which to say that you have a single entity that is an individual human being. They say, sure, the genes are established in the embryo at day one, but you could derive many other embryos from the single embryo as it grows. So that here you have an 8-celled embryo. Each blastomere of that 8-celled embryo could be taken out, plucked out, and could itself be allowed to grow as a human embryo. So the argument is clearly you don't have one distinct individual yet at this time, at the first day, if there is this possibility, the potential for developing multiple embryos. In fact, they would argue, and they do argue, that embryo polarity is not fixed at the time of fertilization. You can see from this drawing, for instance, that there's no north-south axis that you can see in any of these. And you only find the hint of that in the expanded blastocyst later on. This is from research carried out in Davor Solter's lab. He was in Munich, Germany, and I understand he's really recently gone to Australia, but he's considered a major distinguished embryologist. And this is a quote from an article in which he was interviewed, and he says it makes no biological sense to claim that one blastomere, the very first cell, say, always forms the inner cell mass, or even any of those blastomeres in the 8-cell embryo. It makes no sense to claim that one of those always makes the inner cell mass and another the trophoblast. He said that just does not happen. It just doesn't add up. The 14-day or later view goes on to say at about 14 days after fertilization, twinning is no longer possible. Twinning is possible up till then, so that you could start out with an embryo with a certain genetic code, and that embryo could divide and become twins or triplets, whatever you have, for various reasons, up until the 14-day period, so that it's only at that point that you have some assurance that you have some sort of individual entity present. The primitive streak, which is the predecessor of the central nervous system, develops at this time. You've also had the notion that there is the development of what's called the some call it the pre-embryo before this, and call what develops at this point the full-fledged embryo. because the formed embryo separates off from the matter that supports it, the placental material, etc. And you don't have a clear separation of those until around this time. Here you see the primitive street taking shape. That's in the middle picture there, and the primitive groove beginning to appear. That's in the lower photo there. Here you have the twinning argument that I've been referring to. In addition, not only can twinning take place, but you could have two blastocysts that could fuse into one before the 14-day point, so that two embryos may become a single embryo. So the people who take this position say, therefore, before 14 days, there's no individual integrated organism present. And it's only at this later point that you have either an actual entity, actual human being, or some would argue a potential human being. They would say even at the 14-day point, you don't have an actualized being. It has the potential to become one. If you develop it and it develops in certain ways, it could become one. And of course, there's a great deal of disagreement about, if you take the potential view, when it would become actualized. Does it have to have, brain waves? Does it have to have certain organs of the human body? There are a variety of positions that have been taken on this. A third view we'll call the clump of cells view. And basically, it looks at the 4 or 5 days after fertilization, when you would take the embryonic stem cells from the inner cell mass, and it says, look, at this time, you don't find the kind of complexity that you find in human organisms in this 4 to 5 day old embryo. Each is a collection of primitive and undifferentiated cells. And they don't have the potential to develop into human organisms on their own. They depend on external factors that surround them. So they say, much as it's right to use a collection of tissues, such as a kidney, to treat those with serious diseases, so it's right to use a collection of tissues, such as the early embryo, to treat those with serious diseases as well. Now, finally, there's the person view. And the person view says, look, you scientists have fun with all of your theories and all of your observations. You're all a bunch of speciesists. And what you're trying to do is figure out when you have an individual human being present. But they say, if you look at some primates, if you look at bonobos, for instance, they are every bit as advanced in many respects as some human beings are, and we ought to treat them with respect. They become what they call persons because they exhibit the characteristics of a person. That is, they can suffer, they have the capacity for conscious experience, and we shouldn't restrict this just to human beings. And some human beings are not persons, and therefore are not owed the protections that we give to those who are persons. I won't go into much more detail. You may have read Peter Singer. He's one philosopher, for instance, who would take this position, who has taken this position. And so people with this view say we don't owe the early embryo, the early human embryo, protection from destruction because it just is not a person. It has no conscious experience, can't suffer, has no sensibilities whatsoever. Now, this leaves us then looking at where does this stand in terms of what we might do in the future. We want to respect people who believe that the early embryo, respect their view, is a human being from day one if we possibly can. Are there various ways to develop embryonic stem cells that don't involve the use of human embryos? And I don't have time to go into all the alternatives that have been developed, but let me talk about one or two. The most recent one that we've talked about, basically this is what I've been saying, we want to try to produce human embryonic-like stem cells that don't involve destroying human embryos, if we can. And this is the notion of induced pluripotent cells that are derived from skin cells. And this is a picture by Yamanaka, a Japanese researcher. He discovered or developed these stem cells. And at the same time, Jamie Thompson and his colleagues at the University of Wisconsin also published, and they too had developed iPS cells. And what they did was they took fibroblasts, or shortcut adult skin cells, and then induced them to change into embryonic-like cells by inserting 4 genetic factors into them. They used viral vectors to do this. And what you see here is an adult skin cell that a grad student from Toronto very kindly sent me a slide of. And you induce these skin cells to develop into induced pluripotent stem cells. And this is what they look like, vastly magnified. This is one such iPS cell. Now, clearly there are advantages to these cells in the eyes of people who object to the use of human embryonic stem cells, because you don't need to destroy embryos to obtain these pluripotent stem cells. By the way, pluripotent means that they can differentiate into a whole slew of different kinds of cells. Adult stem cells, it is thought, are limited in their trajectory and can only become a limited number of different kinds of stem cells. But pluripotent stem cells can develop into a whole host of multiple different kinds of stem cells. And there is no need to carry out research cloning to obtain these patient-specific stem cells. I'll talk about cloning in a second. But what you get from iPS cells are cells that are genetically the same as the person whose adult skin cell was used, so that if you wanted to derive stem cells for treatments for that individual, all you'd have to do, if this really worked, would be to flake off a skin cell from this person and treat it with these viral vectors that insert the genes, and then start working with the resulting iPS cells and differentiate them into the kinds of cells that the individual patient from whom the adult skin cell was taken. So this would be marvelous if the whole thing worked. However, right now, we know that the genes that are inserted are associated with cancer, at least one of them, and they use viral vectors, which are also associated with cancer. And they've tried it using genes that are not associated with cancer, and they seem to be making some progress along that front. Moreover, right now, iPS cells have not been shown to be efficacious in treatment. And there is concern that the skin cells that are used might be altered by aging or various toxins, so they might create unpredictable risks. And finally, you need millions of lines to develop individualized iPS cells for all patients. So these are some of the advantages and different disadvantages of the use of these. Now, I wanted to move to talk to you a little bit about cloning, because this is another alternative to using human embryonic stem cells, some would argue, because basically some argue that what you get when you produce a clone is not an embryo, but what Paul McHugh calls a clonote, because he says that genetically this is somewhat different from an embryonic stem cell. But if you look closely at the way this works, you take a cell from a patient, and you take an egg from a woman who has donated and you remove the inner cell nucleus of the patient cell, and you insert the donor egg, or you fuse the donor egg and the patient's cell. You zap them with electricity or with chemicals, and when they fuse together, what you get is an individual embryo. And the thought is that this works very well, and you can develop stem cells in this way. And you may remember that a South Korean researcher claimed that he had done this with human beings. He had created human clones and had successfully produced embryonic stem cells from these clones. However, as you probably also know, this was shown to be fraudulent, and there has not been a successfully documented development of human embryonic stem cell lines or pluripotent stem cell lines from human clones to date. There's a company in California, I think it is Stemogen, that claims just in the last month that it has cloned some human embryos. But to the best of my knowledge, they have not developed human embryonic stem cells from these clones. What they have done fairly recently, in November, I think it was, of 2007, they were able to clone monkey embryonic stem cells. And monkeys are mammals, and they figure if we can do it in monkeys, we can do it sometime in humans. So these are two alternative ways of deriving human embryonic stem cells or pluripotent stem cells. There are several others, and we can talk about them later if you would like, but right now I'm going to stick to my time limits and go on and talk to you about a recent development. People are very much interested in deriving patient-specific stem cells. And one of the things they tried recently in the United Kingdom was to insert human cells into cow embryos, or into cow eggs, basically, producing a hybrid embryo, so to speak. Here's a picture of a cow. This is not necessarily the being that would result from this. The concern is that the resulting embryos would somehow emerge as living creatures that would have human features. And they are very concerned. There have been studies that were done in the past that weren't quite the same technique, where they merged embryos of chicks and quails. And so you had chicks that made squawking sounds like quails. And the picture that they envision is that they're going to somehow do this with humans and get humans who perhaps moo like cows. And this is of concern. People are concerned that it would violate human dignity to do this. And the question is, would such studies in some way denigrate human beings if they were to produce these results? Now, the concern is not just that they would look bad, but that they might think and feel like human beings, but be locked into a non-human body. So that with the case of cow-human hybrids, might you have a human being locked into a cow body, thinking like a human being? Do you remember the story of Kafka? What was it when a man was turned into a cockroach and he was locked in? I'm sorry, what was it? That's right, Gregor Samsa. Well, that's the sort of concern that people have here, that humans would desperately be trying to get out of the bodies of animals. There were some studies done in San Diego where human fetal stem cells, fetal cells, they weren't stem cells, I believe, were inserted into, I know what it was human stem cells into mouse fetuses. And they were concerned that these mice would emerge with human brains, and the study showed that there were very few human cells inside the brains of the mice. But, you know, this kind of picture also is in the back of people's minds. You know, where would this take us? So the questions that we're left with regard to this creation of these human hybrids, these human, non-human chimeras, is how do we go about drawing limits, and who should decide this? The National Academy of Sciences has developed guidelines that address the creation of certain kinds of chimeras, and they prohibit certain research, but the idea of using human cow embryos perhaps raises some new questions that aren't addressed by those guidelines. And in the United States, we do not currently have any agency that is directed to regulate this sort of study. And I'll just leave it there. We can talk a little more about that in discussion, but my time has come to an end. Let me stop here and say thanks a lot.